Namaste, everyone, and welcome to another Essential Deep Dive.
Hello.
Today, we're getting into a topic that's, well, absolutely key for understanding life itself and really fundamental for your studies.
That's right.
We're talking about heredity.
Okay.
It's core for class 9th, 10th, 11th, 12th boards, and honestly, it's bedrock material for exams like NEET, UPSC, even state PSEs. A real cornerstone of biology.
Welcome, everyone, to this important deep dive on heredity. You've hit the nail on the head. This isn't just abstract theory. It's knowledge you'll definitely apply. In this session, we'll be systematically unpacking how traits get passed down, generation after generation. We'll look at why we see variations, even within families. And we'll really focus on grasping the foundational rules of inheritance. That includes Gregor Mendel's, revolutionary insights and the fascinating process of sex determination. We'll break down the complex bits step by step. Our goal is for you to not just memorize this for exams, but to genuinely understand the elegance of genetics. So, let's get started.
Let's jump right in. It's something we all notice, right? Children look like their parents, sometimes strikingly so, but they're never identical copies. There's always that mix of similarity and subtle difference. So how exactly does reproduction itself create these variations? Where do the differences actually come from?
That's the perfect place to start our exploration. The process of reproduction, whether it's asexual or sexual, isn't just about making new individuals. Critically, it's also about introducing these subtle differences, what we call variations. These variations, they're the raw material, the fuel for adaptation, for evolution, the basis of all biological diversity, really. Let's think about asexual reproduction first. Here, you have a single parent producing offspring that are essentially clones, like a bacterium dividing or maybe a plant grown from a cutting. You'd expect them to be absolutely identical, wouldn't you? But actually, even here, tiny variations manage to creep in.
How does that happen if it's just cloning?
It comes down to the DNA copying process. It's incredibly accurate, remarkably so, but it's not 100% perfect. Never absolutely flawless. Think about making photocopies. If you copy a copy, then copy that copy again and again. Eventually, tiny flaws appear, right? Little smudges, maybe faded bits. It's similar with DNA replication during cell division. Very small, random errors, inaccuracies can happen in the sequence of nucleotides. These tiny, spontaneous changes in the DNA sequence cause subtle differences in the daughter cells. So take a field of sugarcane, for instance. It often reproduces asexually, and it looks incredibly uniform. But if you could analyze it very closely, you'd find tiny variations between plants, all thanks to these minute DNA copying mistakes. It's the ultimate source, even here.
Wow, okay. So even in something seemingly straightforward like bacteria dividing, which we usually think of as just making perfect copies, these tiny, almost invisible differences are building up over time. That's quite surprising.
Precisely. Let's picture it. A single bacterium divides, making two daughter cells. They're almost identical to the parent and to each other, but maybe not perfectly identical because of those tiny copying inaccuracies we discussed. Now imagine each of those two divides again. Now you've got 4 bacteria in the next generation. Each of these 4 will have inherited some of the minor differences from the grandparent cell, plus they'll have their own newly created differences from their immediate parent cells. So generation after generation, these small inaccuracies accumulate. The basic body design stays the same, they're all still bacteria, but the individual bacteria slowly become more distinct from one another. This is exactly what diagrams like figure 8.1 in your textbooks try to show. An original organism branches out, and each new individual carries inherited differences and its own unique new variations. It's a slow accumulation, but it builds diversity.
That makes a lot of sense for asexual reproduction, fast cloning, small errors adding up. But you mentioned earlier that sexual reproduction somehow maximizes successful variations. How does that work? What's so different there?
Now that's where sexual reproduction really shifts things into high gear in terms of generating diversity. Asexual reproduction mainly relies on those random DNA copying errors for variation. Sexual reproduction, on the other hand, inherently involves mixing genetic material from 2 different parents. This isn't just a small tweak, it's like a massive shuffling of the genetic deck. You're not just dealing with tiny copying errors, you're combining 2 distinct sets of genes, shuffling and recombining them in completely new ways with every single offspring. Think of it this way. Asexual reproduction is like shuffling one deck of cards, and maybe occasionally a card gets a tiny crease. Sexual reproduction is like taking 2 completely different decks, shuffling them thoroughly together, and dealing a unique hand every single time.
Okay, so the potential for new combinations is just vastly greater.
Exactly. That's why if you look at that sugarcane field, again, mostly asexual, you see uniformity. But then look at humans or dogs or cats, organisms that reproduce sexually. You see immense, obvious variation among individuals. Even brothers and sisters, unless they're identical twins, look quite different even with the same parents. And identical twins, starting with the same genes, still develop subtle differences due to environment. So sexual reproduction provides a much larger, faster, and more robust pool of genetic possibilities. It dramatically increases the odds that new, potentially useful variations will pop up in a population.
That's a really clear difference, the sheer scale of variation, and it connects to what you said about survival advantage. These are subtle differences, you said, but they can be really significant for survival, especially if the environment changes. Can you give some real-world examples, maybe beyond bacteria, of how these variations become so crucial?
Absolutely. And this concept is fundamental. It links directly to natural selection and evolution, topics you'll definitely dig deeper into for competitive exams. You're right, not all variations have the same survival value. Some, even seemingly minor ones, can give a huge edge in a specific situation. Let's go back to bacteria, but push it further. Imagine a population doing well in a nice, stable environment. Suddenly, there's a heat wave, or maybe a new antibiotic gets introduced into their environment. Now, if some bacteria, just by random chance, happen to have a genetic variation that lets them tolerate higher heat, or maybe resist that antibiotic, they'll survive and reproduce. The ones without that variation might die off.
Like antibiotic resistance we hear so much about.
Exactly. Antibiotic resistance is a perfect and frankly worrying real-world example. A tiny genetic change, maybe altering a protein so the antibiotic can't attach, gives that bacterium a massive survival advantage when the drug is present. That resistant strain then multiplies, leading to the evolution of superbugs. It's variation plus selection in action. We see it in humans too. Think about the gene for sickle cell anemia. Having 2 copies of the sickle cell gene causes a serious blood disorder. But having just 1 copy, being heterozygous, often provides some resistance to malaria. So in places where malaria is common, this genetic variation, even though it has a potential downside, offers a significant survival advantage. People carrying 1 copy are more likely to survive malaria, reproduce, and pass on that gene.
So the environment is essentially selecting for that trait, even if it's complex.
Precisely. The environment acts like a filter. Another classic example is the peppered moths in England during the Industrial Revolution. Originally, most were light-colored, camouflaged against lichen on trees. Then pollution blackened the trees with soot. A rare, dark-colored variant suddenly had better camouflage. Birds ate the more visible light moths, so the dark moths survived better, reproduced more, and the dark form became much more common. Later, when pollution controls cleaned up the air and trees, the trend reversed. It shows how environmental pressure directly selects for advantageous variations. So yes, these subtle differences can absolutely be life or death, and understanding them is key to understanding adaptation and evolution itself. That's why variation is such a core concept in biology.
That really drives home the impact of variation. It's not just cosmetic, it's about survival. Okay, so building on that, let's focus on how these characteristics actually get passed down reliably. What exactly do we mean by heredity, and how do we balance the similarities and differences we see?
Good transition. Heredity, at its core, is the process by which these traits and characteristics are consistently passed from parents to offspring. The most obvious result of reproduction, as you noted, is creating individuals with a similar basic body design. A human baby is unmistakably human, right? 2 eyes, a nose, limbs. These fundamental features are reliably inherited. They define the species. But within that shared blueprint, there's enormous variation. A child has all the basic human features, but they don't look exactly like their parents or like any other unrelated human. These are the differences in inheritance. Things like hair color, eye shape, height, maybe even predispositions for certain things. These make each individual unique, even while sharing the species-defining similarities. So heredity covers both the reliable passing on of common traits and the mechanisms that allow for individual variation.
Okay, so the species-level similarities versus the individual-level differences. That makes sense. I remember Activity 8.1 in the textbook uses earlobe, something simple, to demonstrate this. How can such an ordinary feature help us understand inheritance?
It might seem simple, but the earlobe trait is actually a brilliant example. It's easily observable and nicely demonstrates basic Mendelian inheritance in humans. If you just look around at your family, your classmates, you'll generally see 2 main types of earlobes. First, there are free earlobes. The bottom part hangs down, sort of detached from the side of the head. You could probably slip a finger behind the lowest part. Figure 8.2 usually shows this clearly. Then you have attached earlobes. Here, the lobe seems to connect directly to the side of the head without that free hanging part. It looks more fused. Activity 8.1 basically asks you to do a small survey. Look at everyone's earlobes in class, count how many have free versus attached, and maybe calculate percentages. But the really interesting part is correlating this with their parents' earlobes. What type do the parents have?
So you start seeing patterns.
Exactly. You might notice, for example, that parents with attached earlobes usually have children with attached earlobes. But what if two parents both have free earlobes, but they have a child with attached earlobes?
Okay, yeah, that would be puzzling at first glance.
Right. That observation, even without knowing the terms dominant or recessive yet, strongly hints that there are underlying rules. It suggests some traits can be carried but hidden, only to reappear later. It's basic data collection leading to a hypothesis, just like Mendel did.
That's a neat demonstration. But for exams, how might this earlobe example be used? What's the practical takeaway for, say, a NEET aspirant beyond just observing earlobes?
Very relevant question for exam prep. On a board exam, you might get a question like, a husband and wife both have free earlobes, but their first child has attached earlobes. Explain how this is possible using genetic principles.
Right.
Your answer would need to explain dominance and recessiveness. Free earlobes are dominant. Let's use F. Attached are recessive. The parents, despite looking like they have free earlobes, phenotype, must both be carriers of the recessive allele. Their genotype must be heterozygous, FF. The child could then inherit the F allele from the mother and the F allele from the father, resulting in an FF genotype, which expresses the attached earlobe phenotype. That tests your grasp of basic Mendelian logic.
Okay, so applying the dominant recessive concept, what about NEET?
For NEET, it could be more involved. You might get a pedigree chart showing earlobe types across generations and have to deduce the inheritance pattern, autosomal dominant or recessive, or you might be given parental genotypes like FFXFF and asked to calculate the probability of their child having free or attached earlobes, likely involving a Punnett square. The real-world application isn't about earlobes themselves, they're harmless variations, it's about the principle. Understanding how a hidden recessive trait works is crucial for things like genetic counseling for inherited diseases. Many genetic disorders are recessive. Parents who are carriers, heterozygous, might show no symptoms, but they still have a 25% chance with each pregnancy of having an affected child who inherits two recessive alleles. The earlobe example helps you grasp that fundamental mechanism, which applies to much more serious conditions and even to breeding better crops or animals. The principle is key.
Got it. It's the underlying logic that matters. And speaking of the logic, that brings us straight to Gregor Mendel, the father of genetics, really. His work laid the foundation. What made his approach so different, so revolutionary, especially since he didn't even know about DNA?
Mendel. His work in the mid-1800s was truly transformative. What set him apart from others studying inheritance back then was his incredibly methodical and, crucially, quantitative approach. He used math. Before Mendel, the common idea was blending inheritance traits, just mix like paint. Mendel wasn't satisfied with just observing. He designed careful experiments, controlled his crosses meticulously, and, most importantly, he counted. He counted the exact number of offspring showing each specific trait in every generation. This focus on numerical data on ratios was the breakthrough. It allowed him to see patterns, statistical regularities that others had missed. He deduced abstract laws purely from these numbers, long before genes or chromosomes were discovered. It was a brilliant fusion of biology and mathematics.
That focus on counting and ratios makes sense. And he started with a basic idea about how parents contribute, right?
Yes. One of his fundamental insights, which seems obvious now but wasn't then, was that both parents contribute essentially equal amounts of genetic information to the offspring. We now know this is DNA. This means for pretty much every trait, height, seed color, earlobes, a child gets two sets of instructions, two versions of the genetic information. One version comes via the father's sperm, the other via the mother's egg. So a child's height isn't just from one parent, it's influenced by the height-related genetic info from both. The final expressed trait comes from the interplay between these two inherited sets of information. This idea of two copies, two factors per trait, is central to everything Mendel discovered.
And he famously used pea plants, Pisum sativum, for these experiments. Why peas? What was special about them or his setup?
An excellent choice, really. Pea plants were almost perfect for his work for several reasons. One, they're easy to grow and manage, and they produce lots of offspring quickly. Short generation time is key for studying inheritance over multiple generations. Two, they naturally self-pollinate. This allowed Mendel to create pure breeding lines, plants that reliably produced offspring identical to themselves for a specific trait, like always tall or always short. This gave him a stable starting point. 3, although they self-pollinate, he could also easily cross-pollinate them manually. He could control exactly which plants bred with which. By transferring pollen carefully and preventing self-pollination, precision was vital. And maybe most importantly, 4, pea plants have several distinct, easily identifiable contrasting traits. Things that are clearly one thing or the other with no confusing in-betweens.
Like tall or short, round or wrinkled.
Exactly. Round or wrinkled seeds, yellow or green seeds, violet or white flowers, tall or short stems. These clear-cut alternatives made counting and tracking straightforward. His setup was rigorous. He'd start with 2 pure breeding parents showing contrasting forms of one trait, like pure tall versus pure short. He'd cross them, the P generation, then observe and count the traits in their offspring, the F1 generation. Then he'd let the F1 self-pollinate and count the traits in their offspring, the F2 generation, and sometimes even further. Methodical, quantitative, brilliant.
Okay, let's break down that first big experiment, the monohybrid cross, focusing on just one trait, like height. What did he do and what did he find in that first F1 generation?
Right, the monohybrid cross. This is foundational. Let's walk through it carefully. Mendel selected his pure breeding plants. A pure breeding ball one, always producing tall offspring, and a pure breeding short one, always producing short offspring. These are his parental P generation. He then cross-pollinated them, pollen from the tall onto the short, and vice versa. The seeds from this cross grew into the first filial F1 generation. The result was striking and flew in the face of blending theory. Every single plant in the F1 generation was tall.
All of them. No short ones at all. No medium ones.
None whatsoever. No medium height, no blending, and definitely no short plants. It was as if the trait for shortness had simply disappeared. This immediately told him something crucial. One trait could completely mask the other.
Fascinating. The shortness was hidden somehow. But then you said it reappeared in the next generation, the F2, when the F1 plants self-pollinated. That must have been the key moment.
Absolutely. The F2 generation held the key. This is where Mendel's careful counting revealed the underlying pattern. He took those F1 tall plants, which resulted from the tall X short cross, and allowed them to self-pollinate. Pollen from an F1 plant fertilized the ovules of the same plant. He collected the seeds, planted them, and grew the second filial F2 generation. And here's what he found. The F2 plants were not all tall. Instead, he consistently observed that about 3/4 of the F2 plants were tall and about 1/4 were short.
Ah, the famous 3.1 ratio.
Exactly. That consistent 3.1 ratio, tall-short, in the F2 was the critical observation. The fact that the short trait reappeared proved it hadn't been destroyed or blended away in the F1 generation. It must have been carried, hidden by those tall F1 plants.
So, inherited but not expressed. This is where dominant and recessive comes in, right? This is super important for Neaton boards. Could you maybe slow down and explain exactly how Mendel interpreted this using factors or genes and the idea of dominant versus recessive? Use the T and T notation.
Yes, let's definitely slow down and make this crystal clear. This is the absolute core of Mendelian genetics. Let's take this step by step. Mendel reasoned from his F1 and F2 results that traits are controlled by heritable units, which he called factors. We now call them genes. He proposed that for each trait, an organism inherits 2 factors, one from each parent. These factors can be the same or they can be different versions. We now call these different versions alleles. Now, let's apply this to the tall-short example. Step 1. Parental P generation. The pure breeding tall parent must have 2 identical factors for tallness. Let's represent the factor for tallness with a capital T. So its genetic makeup, its genotype is TT. The pure breeding short parent must have 2 identical factors for shortness. Let's use a lowercase t. Its genotype is TP, step 2, F1 generation. When TT is crossed with TT, the TT parent can only pass on a T factor in its gametes, pollen ovules. The pet parent can only pass on a T factor. So all offspring in the F1 generation must inherit 1 T and 1 T. Their genotype is TT. Now, critically, Mendel observed that all these F1 TT plants were tall. Their observable trait, their phenotype, was tall. Step 3, interpretation dominant and recessive. This observation that T-type plants are tall is key. It means the T factor masks or overrides the T factor when both are present. So Mendel defined a dominant allele T, an allele that expresses its trait even when only 1 copy is present. Tallness T is dominant. Genotypes TT and TT both result in a tall phenotype, recessive allele. An allele whose trait is only expressed when 2 copies are present and the dominant allele is absent. Shortness T is recessive. Only the genotype CIT results in a short phenotype. Step 4, F2 generation. The F1 plants, genotyped TT, self-pollinate. A T-type plant produces two types of gametes in equal amounts, half carry T, half carry T. Now think about the possible combinations when these gametes fuse randomly during self-pollination. Sperm T plus egg T contains only TT genotype, phenotype tall because T is dominant. Sperm T plus egg T plus egg T plus egg T, phenotype tall, same as TT. Sperm T plus egg T phenotype short, only way to be short. Step 5, the ratios. If you look at the genotypes, you have 1 TT, 2 TT type writer, 1 TT. That's the 1.2.1 genotypic ratio. If you look at the phenotypes, you have TT plus TT, which are all L tall, L and T just short. That gives you 3 tall, 1 short. That's the 3.1 phenotypic ratio that Mendel observed. So this model perfectly explains his results. Understanding dominant recessive, genotype of phenotype, and these ratios is absolutely vital for your exams. Activities like 8.2 and diagrams like figure 8.3 help visualize this.
That step-by-step breakdown makes it incredibly clear. The dominant allele masks the recessive one, unless you have 2 copies of the recessive.
Okay.
But Mendel didn't stop there, did he? He looked at 2 traits at once, the dihybrid cross. How did that experiment, maybe with seed shape and color, reveal his next big idea, independent inheritance, shown in figure 8.5? This sounds really important for NEET, too.
You're absolutely right. The dihybrid cross was the next logical step and led to Mendel's second law, the law of independent assortment. It's definitely a step up in complexity, but crucial. So for this, Mendel crossed plants that differed in 2 characteristics simultaneously. A classic example is crossing a plant that was pure breeding for round yellow seeds with one pure breeding for wrinkled green seeds. From his monohybrid crosses, he already knew round R was dominant over wrinkled R, and yellow Y was dominant over green Y. So the genotypes of his P generation were RRYY, round yellow, and res, wrinkled green.
Okay, pure breeding for both traits.
Correct. He crossed these RRY and Rhodia plants. What do you think the F1 generation looked like?
Well, based on dominance, they should all show the dominant traits.
Yeah.
So all round and yellow.
Exactly. All F1 plants had the phenotype round and yellow seeds. This confirmed the dominance. Their genotype would have to be heterozygous for both traits, RRY. They received R and Y from one parent and R and Y from the other. Now, the crucial part, just like before, was the F2 generation. Mendel let these F1 plants, RRYY, self-pollinate. Think about it. If the traits were somehow linked, if R always stayed with Y and R always stayed with Y, then in the F2, you'd only expect to see the original parental combinations, round, yellow, and wrinkled green, probably in a 3.1 ratio again.
That's not what he found, was it?
Not at all. He found the 2 parental types, yes, but he also found brand new combinations that weren't present in the P or F1 generations. He found plants with round green seeds and plants with wrinkled yellow seeds. And again, his careful counting revealed a consistent pattern, a 9.3.3.1 phenotypic ratio. That's 9 round yellow, 3 round green, 3 wrinkled yellow, 1 wrinkled green.
Wow, so the traits for shape and color weren't inherited as a package deal. They sorted out independently.
Precisely. That's the principle of independent inheritance, or independent assortment. It means the alleles for one trait, like seed shape RR, segregate into gametes independently of the alleles for another trait, like seed color Y, assuming the genes are on different chromosomes or far apart on the same one. Let's think about the gametes produced by the F1 plant, RRYY. Because the RR pair assorts independently from the Y pair during meiosis, which we'll discuss more later, an RRY plant can produce 4 types of gametes in equal proportions. 1, RY, carrying dominant alleles for both. 2, Ry, carrying dominant for shape, recessive for color. 3, RY, carrying recessive for shape, dominant for color. 4, RY, carrying recessive alleles for both. When these 4 types of pollen randomly fertilize the 4 types of egg cells during self-pollination, you get all the combinations that lead to that 9.3.3.1 ratio in the F2 offspring. Figure 8.5 illustrates this beautifully, showing how these combinations arise. This independent shuffling is a major source of genetic variation in sexually reproducing organisms.
That's a huge concept. The ability to predict these outcomes using Punnett squares for dihybrid crosses must be a key skill, especially for NEET. Can we quickly revisit the inherited but not expressed idea with maybe a simple analogy? And then could you just briefly touch on how we'd read a Punnett square for this dihybrid cross, maybe just 1 or 2 boxes?
Good idea. Solidifying these is key. For the inherited but not expressed or recessive idea, let's use your phone's music apps again. Imagine your phone can have apps for rock music, R, which is the default or dominant choice, and pop music, which only plays if rock isn't chosen. A phone with RR genotype plays rock. A phone with PP plays pop. Now, a phone with genotype RP has both apps inherited. But because rock R is dominant, the phone only plays rock music. The pop app is still there, it's inherited, but its function isn't expressed because the rock app dominates. The pop trait is hidden, recessive, only showing up if the phone gets two pop apps. Makes sense.
Yes, that analogy really helps visualize the hidden recessive trait. Okay, now for the dihybrid Punnett square, the 4x4 grid for RRY x RRYY, how do we read it?
Right, the Punnett square is just a tool to organize the random combinations of gametes. For the RRYX RRYY, you list the 4 possible gametes from one parent, RYY RYY, along the top, and the 4 from the other parent down the side. Then you fill in the 16 boxes by combining the alleles. Let's look at just 2 boxes. Top left box combines gamete RY from top with gamete RY from side. The genotype is RRY phenotype. Round, yellow, both dominant traits expressed. A middle box. Maybe combine gamete RY from top with gamete RY from side. The genotype is RRYY, phenotype, still round, yellow, because R is dominant over R and Y is dominant over Y. Another box. Combine RY top with RY side. Genotype, RRY, phenotype, round RR but green, YY. A new combination, bottom right box. Combine RY top with RY side. Genotype, RY, phenotype. Wrinkled green, both recessive traits expressed. By filling all 16 boxes and tallying the phenotypes, you'll arrive at that 9.3.3.1 ratio. The key is correctly identifying the gametes and then combining them systematically. Practice is essential here for speed and accuracy in exams.
Okay, that clarifies the Punnett square application. Perfect. Now, we've talked a lot about how traits are inherited following these patterns, but let's get down to the molecular level. How do these genes, these bits of DNA, actually cause a trait like tallness or round seeds to be expressed? What's the link?
Excellent question. This connects Mendel's abstract factors to the actual chemistry of life. It's all about proteins. Fundamentally, the DNA in our cells holds the master code, the instructions. Think of it like a huge library of blueprints. Cellular DNA is the primary source of information for making proteins, and proteins are the molecules that do almost everything in the cell. They form structures, transport molecules, act as signals, and crucially, they act as enzymes. Now, a gene is defined as a specific segment of that DNA that contains the complete set of instructions for building one particular protein. So one gene typically codes for one protein.
Okay, gene equals protein recipe. But how does a protein then lead to, say, a plant being tall? What's the connection between the protein and the visible trait?
That's the crucial link. Let's use the tall, short plant example again. Plant height is influenced by growth hormones. The amount of growth hormone produced plays a big role in how tall the plant gets. Now, the production of that hormone is a biochemical process. And what drives biochemical processes in cells? Enzymes. Remember, enzymes are proteins that catalyze specific reactions. So here's the likely chain of events. 1, there's a gene, let's call it the height gene, that holds the code for making a specific enzyme. 2, this enzyme is needed for synthesizing the plant growth hormone. 3, now consider the dominant allele, T. Let's say the T allele codes for a highly efficient version of this enzyme. 4, an efficient enzyme works well, producing a lot of growth hormone. 5, lots of growth hormone leads to significant cell growth and elongation, resulting in a tall plant.
Okay, that makes sense for the dominant T allele. What about the recessive T allele?
Right. Now, let's say the recessive T allele is a version of the gene that has a slight alteration, a mutation. This altered T's gene might code for an enzyme that is less efficient or maybe even non-functional. A less efficient enzyme will produce less growth hormone. Less growth hormone means less growth stimulation, resulting in a short plant. So you see the connection. Genes control traits by dictating which proteins, like enzymes, are made and how efficiently those proteins function. The proteins then influence physiological processes, like hormone production, that determine the final observable characteristic. DNA protein innate trait, that's the central dogma in action, explaining Mendel's findings at a molecular level. A T plant is tall because even one copy of the T allele produces enough efficient enzyme to make sufficient hormone for tall growth. Only with TLT do you lack that efficient enzyme completely, leading to shortness.
That connects everything beautifully. Genes, efficient enzyme, hormone level trait.
Got it. And this loops back to parents contributing equally. You said body cells have 2 gene sets, but germ cells, sperm egg, must have only one. How does the cell have its gene sets, and what exactly are chromosomes in this picture?
Yes, that having process is critical for maintaining genetic stability across generations in sexual reproduction. Let's recap. Normal body cells, somatic cells, are diploid. They have 2 complete sets of genes organized into structures called chromosomes. One set comes from the mother, one from the father. In humans, that's 23 pairs, or 46 chromosomes in total. Now, if the reproductive cells, the germ cells, sperm and egg, were also diploid, think what would happen at fertilization. A diploid sperm, 2 sets, fusing with the diploid egg, 2 sets, would create a zygote with 4 sets of chromosomes. And the next generation would have 8, then 16. That would get messy fast.
Exactly. The chromosome number would double each generation, leading to genetic chaos. So nature evolved a specialized type of cell division to create haploid germ cells with only one complete set of chromosomes. This process is called meiosis, and chromosomes themselves, think of them as the organizational units of DNA. Instead of one impossibly long DNA strand, the genetic information is packaged into these distinct, manageable structures within the cell nucleus. Humans have 23 types of chromosomes. In diploid cells, we have 2 copies of each type, one maternal, one paternal, forming homologous pairs, except for the sex chromosomes in males XY, which are a mismatched pair.
Okay, so meiosis is the process that makes haploid germ cells from diploid cells. How does it work? It sounds complicated.
It is intricate, but we can break it down. Meiosis involves 2 consecutive cell divisions, meiosis the first and meiosis the second. Let's take this step by step. Before meiosis starts, the cell first duplicates its DNA. So each of the 46 chromosomes now consists of 2 identical sister chromatids joined together. It's still considered 46 chromosomes, but they're doubled. Meiosis the first, the reduction division, this is where the chromosome number is halved. Pairing up, homologous chromosomes, the maternal copy and paternal copy of chromosome 1, for example, find each other and pair up tightly, crossing over. While paired, segments of DNA can be exchanged between the homologous chromosomes. This shuffles alleles, creating new combinations on individual chromosomes, a major source of variation. Independent assortment. The homologous pairs line up at the cell center. Crucially, how they line up is random. The maternal copy of pair 1 might face left, while the paternal copy of pair 2 faces left. This randomness dictates which mix of maternal and paternal chromosomes ends up in each daughter cell. This is the chromosomal basis of Mendel's law of independent assortment. Separation. The homologous chromosomes separate and move to opposite poles. The sister chromatids remain attached. The cell then divides into 2. Each daughter cell now has only 23 chromosomes, 1 from each homologous pair, but each chromosome is still doubled, consists of 2 sister chromatids. These cells are now haploid in terms of chromosome number. Meiosis the second, the equational division. This is much like a normal mitotic division. The 23 doubled chromosomes line up in each of the 2 cells from meiosis the first. This time, the sister chromatids separate and move to opposite poles. Each cell divides again. The final result: 4 haploid cells, each containing 23 single chromosomes. In males, these become 4 sperm cells. In females, it typically results in 1 large egg cell and smaller polar bodies. Each of these 4 gametes is genetically unique due to crossing over and independent assortment.
Wow, okay. 2 divisions. First, separating the homologous pairs, having the number, then separating the sister chromatids. That's much clearer. And if this didn't happen, the chromosome number would just keep doubling. That really emphasizes why meiosis is so vital for species stability.
Absolutely vital. It ensures that when a haploid sperm fertilizes a haploid egg, the resulting zygote restores the correct diploid number, 46 in humans, maintaining the species' characteristic genetic blueprint generation after generation. It's a beautiful mechanism that explains Mendel's laws and ensures both continuity and variation. And while asexually reproducing organisms don't do meiosis, they still pass on traits. Their variation comes mostly from mutations during DNA copying, which are then passed directly to clones. It's efficient for stable environments, but lacks the massive reshuffling power of sexual reproduction.
That clarifies the whole chromosome picture. Okay, moving on to another really interesting aspect tied to chromosomes: sex determination. How is the sex of a baby decided? You mentioned it varies. It's not always the XXXY system we know in humans.
That's right. Sex determination is surprisingly diverse across the living world. It's not a universal system at all. In some species, it's entirely down to environmental factors. Take many turtles and alligators, for example. The temperature at which the eggs are incubated during a specific developmental window actually determines the sex.
Temperature.
Really?
Yes. For instance, in American alligators, eggs incubated around 33 degrees C tend to become males, while those around 30 degrees C tend to become females. It's called temperature-dependent sex determination, or TSD. No specific sex chromosomes involved there.
That's wild. And I think I read somewhere that some animals can even change sex.
Correct again. That's another fascinating strategy called sequential hermaphroditism. Some fish, like clownfish, Finding Nemo, are born male, and the dominant male in a group can become female if the resident female dies. Some other species start as female and change to male. It shows that in these animals, sex isn't fixed genetically from birth, but can be influenced by social or environmental cues later in life. But when we look at humans, it's overwhelmingly genetically determined right at fertilization. The environment doesn't change it afterwards.
So back to the X and Y chromosomes. We know women are XX and men are XY. How does inheriting these lead to determining sex, ensuring a mix of males and females?
It comes down to that one odd pair of chromosomes, the sex chromosomes. Remember, humans have 23 pairs total. 22 pairs are autosomes, same in both sexes. The 23rd pair is the sex chromosomes. Females have 2 identical normal-sized sex chromosomes called X chromosomes, genotype XX. Males have 1 X chromosome and 1 much smaller Y chromosome, genotype XY. The Y chromosome carries the crucial SRY gene that triggers male development. Now think about meiosis and gamete formation. A woman, XX, undergoes meiosis. All her egg cells will receive 1 X chromosome. She can only produce X-carrying eggs. A man, XY, undergoes meiosis. Because of chromosome segregation, half of his sperm cells will receive the X chromosome and the other half will receive the Y chromosome. He produces roughly equal numbers of X-carrying sperm and Y-carrying sperm. Now, fertilization. The offspring always gets an X chromosome from the mother's egg. The sex is determined by the sperm. If an X-carrying sperm fertilizes the egg, zygote is XX, develops into a girl. If a Y-carrying sperm fertilizes the egg, zygote is XY, develops into a boy. Because roughly half the sperm carry X and half carry Y, there's essentially a 50% chance of conceiving a boy and a 50% chance of conceiving a girl with each pregnancy. It's a simple, elegant genetic mechanism. Diagrams like figure 8.6 show this clearly.
So it boils down entirely to whether the father's sperm contributes an X or a Y. The mother always gives an X. Is this specific point that the father determines the sex a common question in board exams or NEET?
Yes, absolutely. You've nailed a key takeaway for exams. In humans, the father's contribution, X or Y sperm, is the sole genetic determinant of the child's sex. And yes, this is a very frequently tested concept in board exams, NEET, UPSC, prelims, you name it. You absolutely need to be able to. 1, explain the XXY mechanism clearly. 2, draw or interpret a Punnett square or diagram, like figure 8.6, showing the cross XX female, XY male, and the resulting offspring probabilities. 3, state explicitly why the male parent determines the sex, because he produces 2 types of gametes regarding sex chromosomes. 4, know the theoretical probability, 50-50. It's a fundamental concept linking chromosomes to a major phenotypic trait. Make sure you have it down cold.
Got it. Crystal clear and definitely noted for revision. Thanks.
You're very welcome. We've certainly covered a lot of essential ground today. Let's just quickly wrap up with the main points from this deep dive on heredity, things you really need to lock in for your exams. First, variation. It arises during reproduction, even asexual, but sexual reproduction maximizes it. These variations are crucial for survival and evolution. Second, Mendel's laws. Traits are controlled by genes, alleles, inherited in pairs. Dominant alleles masked recessive ones. The 3.1 phenotypic ratio in monohybrid F2 reveals this. Third, independent assortment. Alleles for different traits on different chromosomes are inherited independently, leading to new combinations, seen in the dihybrid cross 9.3.3.1 ratio. Fourth, the molecular basis. Genes, DNA, code for proteins, like enzymes, which carry out functions leading to traits. Gene unprotein munch trait. And fifth, sex determination. Diverse strategies exist, but in humans, it's genetic. XX for female, XY for male, determined by whether the father contributes an X or a Y chromosome via his sperm. This chapter, Heredity, is foundational for all the genetics you'll study later, especially for NEET. My advice: revise these concepts thoroughly. Practice those Punnett squares for monohybrid and dihybrid crosses using past year questions. Try applying the principles to new problems. And really understand meiosis and its role in variation and chromosome stability. Don't just memorize, aim to understand the logic. If any part still feels confusing, please don't hesitate to revisit it or seek clarification. Keep up the hard work, keep learning, and keep growing. You're building a strong foundation for success.